Alright, so we're back with part two now of our lecture on the prokaryotes. And as we talked about before, bacteria, prokaryotes in general, cannot reproduce sexually because they can't make sperm cells or egg cells. They're made of one cell only, so by definition, they can't make multiple cells. They do, however, have three ways of exchanging or sharing or transferring genes horizontally within the same generation. And so we're going to spend a few minutes now talking about conjugation, transduction, and transformation. Archaeobacteria can do these same processes, but we're going to focus on these primarily in bacteria. So the first one we're going to look at is conjugation. And conjugation is where two bacteria can exchange plasmids. And we talked before in the previous section about how plasmids are small bits of nucleic acid, genes, that are not essential necessarily for functioning of the bacteria, but may be used from time to time to make some other structures or do some other things, perhaps to carry out some other chemical reactions, if a particular individual has the plasmid. So, for example, E. coli comes in two types. There are two strains. Some of them have what's called the F plasmid, or fertility factor. Other ones do not have the fertility factor. And so, if they're able to do conjugation, then they have the fertility factor. If they're able to exchange plasmids back and forth, those bacteria have to be of the F plus type. The F minus type are the cells that don't contain the plasmid. So what we see over here on this slide are two cells in conjugation, and what you'll notice, the one on the top is relatively smooth on the outer surface. The one on the bottom has these little spikes sticking off it. It almost looks like a really sickly little tiny microscopic sea urchin. But this cell actually has the genes that it needs to make pili. And so this cell would have the ability then to attach to things in its environment, to adhere to things, and perhaps have a better or easier chance of being picked up by a host. So the way that this works is this. Again, this is not vertical gene transfer. This is transfer of genes within a single generation. So this organism over here, this is the F plus type. This will be the type of bacteria which has the plasmid. This is the type down here. I'm sorry, the type over here that does not have the plasmid. And so this organism is going to conjugate with the organism over here. This one has a pilus, which is able also to use to transfer plasmids from one cell to the next. So if these two organisms hook up, if they're able to attach, if this organism with its pili are able to attach to this organism, they sort of come together. Their cell membranes are going to fuse because they're now relatively close together. And so once this plasmid is copied, one copy of it's going to go to this bacteria over here. The original bacteria is also going to maintain a copy of it, and so now both of them have the plasmid. They both have the ability to produce pili. They both have the ability to transfer these plasmids from one cell to the next. And so this picture over here is an actual photograph of the cartoon version we saw before on the other slide, where we see conjugation happening between these two bacteria. Transduction is going to involve viruses. We'll take a look at this one here in a second. Transformation we've seen before when we saw the Griffith experiments. Transformation occurs when a bacteria picks up bits of DNA in its environment, takes them into itself, and begins to express them. Of course, mutation is always an option, but as we've seen before, and hopefully as I've convinced you, mutation is not a reliable source of generating new alleles or new genes in a population because it is extremely rare and because it is also random. You can't count on even the few mutations that do happen on giving you beneficial genes because it is such a random process. So we've seen conjugation. We've seen cells that have the ability to do this. There are cells that have an ability to also do some recombination. So the F plasmid itself, even though it is a plasmid, can actually become part of the bacterial chromosome. So just like in eukaryotes, when we see crossing over between homologous chromosomes during prophase of meiosis I, there is a similar type of event that occurs in bacteria. If bacteria have plasmids, if those plasmids come into contact with the chromosome, they can actually exchange bits of information. So some genes from the plasmid might be incorporated relatively permanently into the chromosome, and the reverse is true as well. Genes from the chromosome may go onto the plasmid side as well. Some cells apparently have more ability to do this than others. We call these HFR cells, or high frequency of recombination. They have what appears to be a higher than average rate of this type of recombination happening through time. There is also the ability of the plasmid to excise itself or cut itself out of the DNA, which is sort of the backwards process where the genes will come back out of the chromosome from time to time. So we actually go to removing the plasmid from the chromosome. And because this is not entirely accurate, at times the plasmid may actually contain bits of what was initially chromosomal DNA. You can imagine sort of doing a cutting and pasting sort of event where instead of cutting exactly the piece that you wanted, you get a little bit on each side of that plasmid when you remove it from the chromosome. So the plasmid, once it's excised, once it comes back out of the chromosome, actually also contains some chromosomal DNA. And if that cell were to conjugate with another cell, it could then transfer that DNA onto the other cell. So this would again be a way of exchanging DNA that is both plasmid DNA as well as from time to time some chromosomal DNA. So it's a different type of entity that we're going to start moving from one cell to the next. When we start talking about chromosomes, those can be essential genes that are being moved from one cell to the next. Okay, so here's what this looks like. We have a chromosome up here, and we have an F plasmid. So this cell is going to incorporate a bit, actually this plasmid, into its chromosome. So what's happening here is we're going to take this cell, this plasmid, and insert it into this chromosome, into this bacterial cell. So eventually you have, again, our HFR cell, which has a chromosome, it has a plasmid, and it has chromosome. And presumably, we're going to assume this didn't happen in the middle of a gene this bacteria needed, because again, this is not necessarily a very accurate process, certainly not a directed process. There's nothing saying this plasmid can't be stuck in the middle of some really important gene, and if that were to happen, the cell would die. So for the purposes of this picture, we're going to assume this plasmid gets stuck into the chromosome in a place that's not going to hinder the cell in any way. All right, so eventually, excision is going to happen. So we have our F plasmid, which can then be excised or cut out of the chromosome if the need should arise. And so we're going to take this plasmid back out again, but in this case, we now have a bit of chromosomal DNA going along with it. So the chromosome loses a little bit of DNA in the process of reforming this plasmid as the plasmid comes back out of the chromosome. All right, so then we have transduction. Transduction is a situation where viruses are going to be involved. So we've seen before how viruses are going to infect their host. They're going to become attached to the host. They're going to penetrate or inject their DNA into the host. The host is then going to start manufacturing viral DNA and viral proteins, eventually assembling those proteins and allowing for release of those viruses out of the cell, the assembled viruses out of the cell, presumably in some type of lytic process. So when that occurs, what we have are viruses that have infected a cell. The DNA is in there. The bacteria is mostly making viral DNA and viral proteins. Eventually assembly occurs, and just as a matter of chance, as a mistake that's made, because again, nobody is trying to make this happen. There's nothing directing this process, but from time to time, when those new viruses are being assembled, we're grabbing viral DNA, we're grabbing viral proteins, we're sticking them together, and we're making new viruses that can be released out of the cell. What happens if during that process a bit of bacterial DNA is grabbed? Instead of viral DNA, we grab some bacterial DNA, package that inside of a protein, and assemble a virus that consists of viral protein and bacterial DNA. If lysis of the host cell occurs, that virus gets released out into the environment. If it has a chance to infect another bacteria, the process is going to start over the same way it did before. So the virus, again containing bacterial DNA, is going to attach and penetrate, eject its DNA into the new host, and cause, now we have a bacteria that's picked up bacterial DNA from another individual. Right? So this is another way of transferring DNA that it's not happening because the bacteria want it to happen. It's happening because the bacteria are being attacked by viruses, and the viruses are not especially completely efficient at what they do. It turns out to be a way bacteria are able to move their DNA around. Specialized transduction happens during the lysogenic cycle. Right? So again, this is an accidental process that happens when, again, you can imagine if you have the viral genome integrated into the bacterial genome, the time comes for induction to occur. That's induction to occur. And so what do we have to do for induction? We have to remove the viral DNA from the bacterial DNA and then start using it to manufacture new viruses or new virions. What happens if when that viral DNA is cut out of the bacterial chromosome, if we take some bacterial DNA along with it? We're going to package up viruses, most of which have mostly viral DNA, but occasionally some of them might contain bacterial DNA. And if those viruses go out and infect another bacteria, now we have sort of a hybrid bacteria. It's mostly the DNA it had before, plus some of the bacteria from the other guy that had kind of some bad luck and was killed by the viral infection. All right, so here's kind of a little image of what this might look like. We have bacteriophage out here on the outside of the cell, attaching, penetrating, injecting DNA in. We've got a bacterial chromosome. And so when that viral DNA is infected, some of the proteins produced by this viral DNA are going to be used to chop up or break down bacterial DNA. So the bacteria can't fight back. They can't continue to make their own proteins. They're essentially hijacked at this point. They have no genetic control of any kind because the viruses have taken them over. And so the viruses are going to start using the machinery of that cell to manufacture more viral components, assemble some new viruses, and eventually cause those viruses to be able to leave the cell. So if we have this little guy over here, which if you notice is purple, what this is a bacteriophage, bacterial protein. The shell of this thing is still viral protein, but it contains bacterial DNA. And so if this bacteria is released from the cell, it goes out, finds the next bacteria, injects the DNA, which is bacterial because it's purple, apparently. And as you know, bacterial DNA is always purple, apparently. We're going to have this integration of the initial bacterial DNA with the DNA of the new host cell. So we get this sort of a hybrid situation with the generalized transduction. Transformation is where, again, bacteria are able to pick up DNA from their environment. So a cell is broken down, it dies, its DNA is released in the environment. If another bacteria happens to come across it and pick it up, that bacteria is able to use those genes. They become permanently integrated into that new bacterial genome. Right? So, well, why does this really matter? These bacteria, well, they're not all the same, are they? They have differences, just like we have differences. They have different alleles, perhaps even different genes as a result of mutations. So that transformation process is one way of spreading those new genes within a population of bacteria. If one cell dies, another one picks up its DNA and then starts to reproduce. If it starts to go through conjugation, or if it goes through transduction, now that DNA can be spread among several different bacteria. So this is all horizontal gene transfer. This one's not accidental. The bacteria are actually gathering up these bits of DNA on purpose. We're not talking about any viruses involved here. This is strictly a bacteria picking up bacterial DNA kind of a process. Right, so this is what artificial transformation looks like. This is essentially what the Griffith experiments did. If you remember all the way back to chapter 14, when we talked about DNA and how we got to know that DNA was the stuff of heredity, this is essentially what was going on. Imagine this is one of those bacteria, the smooth strain bacteria, which were able to cause the infection of the mice, which caused the mice to die. If we heat up those bacteria and kill them, we release their DNA into the environment. We can then take another bacteria, the R strain bacteria, which are the non-virulent type. If this bacteria picks up a piece of DNA which tells it how to be a pathogen, well, now it's transformed into a pathogen. And this is permanent, right? This is not a temporary thing. This is not reversible. Once this happens, this bacteria, now and forever, for the most part, is going to have that bit of DNA that the original cell had. So unless you have some type of maybe a transduction event or something which could accidentally remove this from the chromosome, which you have to think would be relatively unlikely to just grab that bit of DNA, this bacteria is now permanently transformed and it's going to express those genes from here on out. Right? So this is another way to move DNA or move genes around within a population of bacteria. Since they can't do meiosis, since they don't go through fertilization, they have relatively little capacity to have hybrid DNA for the most part. What they have is the ability to transfer DNA and move DNA around within the population and spread new genes that pop up relatively quickly. Why is this important? Well, think about antibiotic resistance. It takes just one bacteria with a mutation for some type of mechanism for resistance against penicillin or amoxicillin or vancomycin or whatever you want to talk about. One cell that has that DNA, if it conjugates, if transduction occurs, if it dies and transforms another bacteria, we're going to start spreading that new gene, that new allele within the population. And before you know it, you've got millions or hundreds of millions of bacteria that have that gene for resistance. It just takes one, right? So even though they can't reproduce sexually, they still get by pretty well using this horizontal gene transfer. So this is the end of section 2. What I'm going to do now is turn this off and set up the third section. This is actually the end of a section that I did on this Camtasia program back in the fall. So I'll have that as file number 3. So hopefully this all goes pretty well. Please come with whatever questions you have about Chapter 28 on Monday, and we will talk more then.